The man-eater of men is an insect, a fly that in the larval stage of its life cycle is an obligate parasite of warm-blooded animals, including man. Its scientific name is Cachlyomayaia amanavorax, but it is commonly known as the screwworm fly. Its survival is dependent on its predatory nature, and as such, it becomes one of the most destructive pests in the American continent. A wound as small as a tick bite is sufficient enough to allow the female fly to begin its offensive, to lay its eggs on the edge of the wound. Eggs hatch hours later, and the tiny larvae penetrate the skin. Larvae abrade the tissue with their oral hooks and feed off the exudation. The odor emitted from the infested wound attracts other flies, which deposit more eggs and form a sort of biological chain reaction. If not controlled in time, the screwworm can multiply fast and expand rapidly not only over the country affected, but also extending its reach over broad geographical areas without regard to international borders. Today, its presence affects countries in South America and some islands in the Caribbean. Having become a public health problem, the destruction caused by this tiny parasite costs the international community more than $3.75 billion annually. In areas with poor sanitary conditions, it is very easy for it to attack large sectors of the population surviving on meager economic resources, principally small children and the elderly. The screwworm also seriously affects the economic well-being of the livestock sector, generating losses in meat and milk production, animal skins, and causing mortality in livestock and wildlife. The screwworm can create undue hardship for countries that minimize or are unaware of its destructive nature. The first reports on this insect date back to the 16th century during the conquest of the Americas. Spanish sea captains told of numerous cases in humans, mainly prisoners and slaves, where larval infestations literally ate them alive. In 1858, in French Guiana, deaths among prisoners in the Devil's Island penal colony increased. According to the descriptions, they died victims of larvae that ate their flesh. It was at this time that a French physician, Charles Coquerel, discovered that the fly was a species different from those known at the time and named it Lucilia amanavorax, which literally means shiny fly that devours men. During the second half of the 20th century, screwworm cases in human beings became more evident. In the United States, the first cases were reported, followed by Puerto Rico and Mexico with isolated cases. But in El Salvador, Nicaragua, and Jamaica, the numbers shot upward, reporting 681 human cases between 1990 and 1999. Nonetheless, the solution had begun to evolve since the 1930s, when a research station was established in Georgia by the United States Department of Agriculture. In the 1930s, Drs. Bushland and Knipling did research on rearing screwworm flies artificially and on its sexual behavior, at which time the idea of biological control to tackle the problem was born. In 1950, Bushland and Hopkins conducted experiments to sterilize the flies, working first with X-rays and later with gamma rays. Their research allowed the United States to initiate an eradication program. In 1965, at the point of eradicating screwworms from Florida and the southwestern United States, an agreement was reached with Mexico to continue eradicating by overflying the northern border states of Mexico. It wasn't until 1972 when another collaborative agreement with a new eradication strategy was signed. a group of men with a vision, and with the support of their respective governments, agreed to create the Mexican-American Commission for the Eradication of Screwworms. Thus it was decided to build a new sterile screwworm fly rearing facility in the municipality of Chiapa de Corzo in the state of Chiapas in southern Mexico. The site for the new plant was chosen for its strategic location, adjacent to the sterile fly biological barrier to be established over the Isthmus of Tehuantepec. The eradication program in southeastern Mexico would continue from this site. The rearing facility was designed to produce over 500 million insects a week. Because the insects are reared under conditions simulating ideal natural environmental conditions, the life cycle of the insect is completed in 21 days. The physical conditions, temperature and humidity, of a wound are reproduced artificially in the plant, which allows the rearing of larvae in a controlled environment. The rearing process begins in the colony with cages containing pupae. Approximately 60,000 pupae are put in each cage, from which some 55,000 flies emerge. After mating and egg development, the insects are taken to the oviposition room, where the flies deposit their eggs on boards that have been placed inside the cages. In this room, environmental conditions are strictly controlled in order to generate maximum egg output from the flies. A biological stimulant is used to get the flies to oviposit. The egg masses are then collected and weighed. The egg masses are separated into 1-gram portions. Each portion is then placed on 3 liters of diet in a tray to initiate the larvae rearing process. Diet composition consists of spray-dried bovine blood, egg and milk substitute powder, a gel base, formaldehyde, and water. 48 hours after initiating the larval rearing process, the trays receive 7 more liters of diet and are moved to a third and last initiation room. 24 hours later, 72 hours after the initial feeding, the rearing trays receive a third and final feeding of 6 liters and are moved to the main rearing floor. Approximately 96 hours after initiating the larval rearing process, larvae, in following their instincts, stop feeding, crawl off the trays, and drop to the floor. They fall into canals, where they are collected and taken to the pupation room. The next step is to weigh the larvae by group. 3 liters of larvae are put in a pan containing 6 liters of sawdust. They are kept there for 24 hours, which gives them ample time to pupate. The newly formed pupae are then passed through a pupae sawdust separator, and only the pupae cross over to the adjacent maturation room, where they are spread out in screen-bottomed trays for maturation. Almost 5 days later, the pupae are put in 4-liter cylinders and passed through the irradiators. By this time, the fly is almost totally developed, and only its reproductive organs are affected by the irradiation. The insects are exposed to cesium-137. The cylinders are emptied, and the pupae are again spread out on perforated trays and moved into a cold room with a temperature of 10 degrees centigrade. At this temperature, the insect's metabolic rate slows down and keeps the flies from emerging. This chilling method allows for better control of the insects. Sufficient numbers are accumulated to meet shipment requirements, and delivery schedules are met. It keeps the flies from emerging during transportation after having been removed from the cold, and narrows their emergence span at distribution centers. Fly dispersion is done with aircraft specially equipped to transport and release chilled flies over geographical areas designated for treatment. Standard dispersal grids have 2-mile swath widths, and the amount of flies dispersed per square mile can vary. These strategies are constantly being reviewed and revised in order to reduce costs and maximize efficiency in the eradication effort. All materials used in the insect production process are tested by QC, Quality Control Department personnel. All products must meet established quality standards before they can be used in production. The sterile insects being produced also must meet strict quality parameters established to guarantee highly competitive insects in the field. QC personnel conduct tests on the irradiated insects to monitor quality and ensure they fall within the specified control limits. Tests include percent emergence, fly agility, fly longevity, with and without food sustenance, sexual aggressiveness, sex ratio, and percent malformations. The Biological Security Department has preventive security measures in place. It follows strict controls for people, equipment, and materials, including the elimination of waste products, entering and leaving the biologically secure area of the plant. At the same time, sterile flies are released around the production facility, and a permanent trap line is maintained, principally to monitor fly distribution as part of the preventive measures in place to ensure biological security. The protection of the environment is very important to the Commission. For this reason, the residual water treatment program consists of a 3-lagoon oxidation system, filtering tanks, ultraviolet rays, and chlorination at the end of the wastewater process to ensure it exceeds standards before being discharged back into the Grijalva River. Composting of solid waste products leaving the plant is another feature of the effort to have a cleaner environment. As a result of the composting work, the lagoon system, and many other systems installed, the Commission received a Clean Industry Certificate. The plant has very high standards of quality and efficiency. The installations have an operational plan which places special importance on preventive and corrective programs, as well as the permanent development of a modernization program in engineering and maintenance areas. Through this plan, an electric power generator was put into operation, which not only satisfies energy demands in case of an energy shortage, but helps regulate consumption, along with other energy-saving projects. These efforts garnered a second-place National Energy Savings Award for the Commission in the year 2001. Since its inception, the Commission managed to bring together and maintain an excellent group of specialists committed to the eradication effort. Veterinarians, entomologists, engineers, aviation pilots, administrative personnel, biologists, equipment operators, and technicians have made substantial progress in eradicating screwworm and have kept the program running successfully. It took 19 years of continuous work to completely eradicate screwworms from Mexico. However, no country can ever really be safe from an infestation so long as there are other countries where the pest exists. As proof of this reality, we have Mexico, where in spite of having eradicated screwworms, there have been 3 outbreaks which have been successfully eradicated since then. The implications from those outbreaks dictate better oversight and inspection controls at airports, border crossings, maritime ports, and animal health stations. One small infested wound in a person or animal, or the transportation of a fertile fly, can be the source that starts an infestation in a country free of screwworms. Since its creation 30 years ago, the Mexican-American Commission for the Eradication of Screwworms has demonstrated that international collaboration has a far-reaching impact in the economic development of participating countries. It also transcends cultural differences and individual interests when scientific and technological progress is shared among nations which could not solve this problem on their own. Unfortunately, in many countries, The true dimensions of the danger to the human population and livestock production have not been thoroughly evaluated. Today, men, women, and children pay the consequences. In areas far from the big cities, hundreds of people are infested with screwworms. These families, without information or other alternatives, resign themselves to losing a loved one and to the reduced herds of livestock upon which they depend for survival, believing that no solutions within their reach exist. Many countries understand the importance of the work undertaken by the Commission and have collaborated over these last 3 decades of international cooperation. They have shared in the achievements, such as the eradication of North and Central America, as well as a dangerous outbreak in Libya, which threatened the entire African continent and could have decimated the greatest wealth of wildlife on the planet. An indisputable fact of the screwworm eradication effort has been its cost-benefit ratio, which has been highly satisfactory when we consider the use of sterile insects for biological control. The Commission has the infrastructure and the manpower necessary to respond immediately, anywhere, anytime there is a threat from screwworms to public health or livestock production. For this reason, the Commission's work will continue to provide concrete results on different latitudes, which instills pride and motivation in those who are part of this international effort. The results of the eradication efforts so far have been very encouraging. Nonetheless, the task has not ended. As in a chess game, the screwworm finds itself at a disadvantage, given the progress made in science and technology to combat it. But the fly can only be checkmated when the international cooperative force is strengthened to the point that all nations still infested have a real opportunity to eradicate it once and for all. In the meantime, the man-eater continues its offensive.